clinical study started? Well, it was because we've been working quite hard for the last year and a half, really, to develop a manufacturing process that would be scalable and would meet FDA's requirements to allow us not only to bring a product into the clinic, but in the event that the product's approved, that we'd be able to make the product available to a large number of patients. And so that has taken quite a bit of work. We've done dozens and dozens of manufacturing runs on autologous, or on stem cells donated by healthy volunteers to make sure that we're able to dial in the manufacturing process, generate a data package, and submit that in our IND, which was actually just cleared over the winter. So once we had our IND cleared, we've moved forward as quickly as possible to opening the system study. And so just to kind of orient you to where we're at in the process now. All right, so moving on from the blue box, once you have your IND cleared, you move into clinical research and development. And traditionally, clinical research and development is comprised of 3 different phases: phase 1, phase 2, and phase 3. In phase 1, you simply expose typically a healthy volunteer to your drug to ensure that the drug is safe before moving into patients that have the disease that you're trying to treat. In phase 2, you then move into patients and you try to understand the dose range of the drug. What is the treatment effect? How should the drug be used? And then in phase 3, you typically run 2 parallel studies side by side to generate data for submission of the drug, what we call a new drug application, to FDA or other health authorities around the world. Now, that's a process that takes anywhere from 2 to 10 years, so it can be quite a protracted process. The good news is that that process is quite a bit different for rare disease, and it's quite a bit different for cell therapy. So I've kind of tried to outline some of the differences there in the bullets below the schematic. But I think the biggest difference to mention for you is that because cell therapy or a gene therapy is a one-time treatment and it's irreversible, we don't expose healthy volunteers to the treatment. So what that means is we have a higher bar before we go into the clinic, but once we go into the clinic, we go directly into patients that have the disease. And effectively what it allows us to do is to combine phase 1 and phase 2 into a single study. The next thing that I think is really important to mention here is that because these treatments are irreversible, we will continue to study these patients for up to 15 years. So I mentioned that clinical development is a low probability of success business. On the other hand, we really are convinced and optimistic about this program, and so we're investing really a commitment to follow patients that we're treating in this study for 15 years. So just to give you a sense of the level of commitment behind moving into the clinic with this study. All right, so let me move to the next slide, and I'm just kind of, this is a nice transition before we start to talk about the system study. So drug development in general, I can show you the schematics of drug development, is that we are trying to understand in development programs the benefits and the risks of a potential new medicine. Really, at the end of the process, we've generated data on both sides, and we have a look at the medicine and try to decide, does the benefit outweigh the risk? Is this a medicine that we should expose patients to? And ultimately, as someone that uses medicine, I'm sure everyone in the room has at one point or another used medicine, that's what you want to know, right? You want to know, how is this going to help me and what risks am I taking by using it? That's what physicians want to know when they're prescribing a medicine to patients, and ultimately that's what FDA wants to know when they make a decision on whether or not a medicine can be sold in the US. And so as we talk about the system program, I want to be able to consistently bring us back to the objective of the program, which is to assess the benefits and risks of DFT-383, which will allow us to move forward with bringing the therapy to more patients. All right. Okay, so I'm going to do this in a couple of steps. I'm going to start off by talking about the objectives of the study, and then I'll introduce you to an idea we call study endpoints. Then we'll talk about eligibility criteria for the study. So what patients are we studying, at least to start off with? And then I'm going to show a schematic. How is the study designed? And then we'll talk about what that actually looks like in practice. And then we'll wrap up. All right. So the objective of the system study of DFT-383 is to assess the safety and effectiveness of the product. So going back to that idea of benefit risk, safety and effectiveness, so the same idea. So when we run a clinical study, we collect, we measure a huge number of things. And so when we look at all this data that we're generating, we need to structure it and prioritize it in some way. So what we do is we prioritize endpoints by, before the study starts, we describe them, just a few endpoints, a few measurements that we're taking in the study, we describe as our primary endpoints. We follow that with some other, the next level of importance of measurements we're taking, which we call secondary endpoints. And then everything else that we're measuring, we call exploratory. And so just to give you a sense of how we structure and prioritize the data we're capturing. Now, what we try to do is we try to make that prioritization, line that prioritization up with the objectives of the study. Basically, what are the most important pieces of information that we are trying to generate on both sides of that balance beam that I just showed you. So for us in this study, the primary endpoints, which you can see in the middle of the slide, are overall safety. So we need to understand we'll be keeping very close track of what are adverse events associated with the treatment for patients. Secondly, we're looking at hematopoietic reconstitution. So we want to see that the stem cell therapy is engrafting and that patients are recovering. And Natalie talked a little bit about what that process looked like for her in the RDO4 study. And then thirdly, we want to see the effect of the treatment on the patient's kidney function. And we've shortened this to say reversal of renal Fanconi syndrome. Now, that's just so that I don't hit you guys with a super complex slide. So when we talk about reversal of renal Fanconi syndrome, there are a number of very specific laboratory measurements that sit behind that. And we will be looking across all of them in what we've designed a rather complex, what we call composite endpoint, where we're looking at a bunch of different measures, and we'll be looking to see basically if there's any improvement in kidney function at all in patients with renal Fanconi syndrome. The secondary endpoints that we're going to be looking at are really focused on first safety, with looking at the overall safety and tolerability of the other steps in the process related to the cell therapy. And then secondary efficacy endpoints, one of which I think you would agree is really important to patients with cystinosis, which is independence from cysteamine. We'll be looking at a cystinosis-specific PRO. PRO is an acronym for patient-reported outcomes, so it is basically a questionnaire. How does a patient feel or function when they're on treatment? And then lastly, we will be looking at tissue-level cystine depletion. And Stephanie showed some nice slides showing the tissue-level cystine depletion that she's seen in the patients in the RDO4 study. You can see there's some of the other exploratory endpoints outlined. I think I won't take time to go through those, but to step back and maybe a couple of last comments about the endpoints that we're studying. And actually, the first thing I want to mention is really, really important, is that success or failure of the primary endpoints at this point in the clinical development of DFT-383 does not equal study failure. And I think that's really important. So I'm actually going to use my back button here, and I'll flip back a couple slides. This is kind of traditional schematic. When we talk about drug development, if you open up Bloomberg and click on any random pharma company's news stream, you'll see a lot of data coming in on phase 3 studies. Now, in phase 3 studies, you have to pre-specify your primary endpoint and you've got to hit it. And if you don't hit it, your program's over, more or less. Okay? When you're in phase 1 or just entering the clinic, you are not yet in that position. So there's still a lot of exploration that goes on. You're collecting a lot of data that you really don't know which direction each one of those measurements, each one of those data points is going to go, because you haven't yet seen the treatment effect of that drug in that population. So it's very, very normal for the structuring of endpoints in a clinical study that's this early to change, right? So I think that's something really important to bring up. This is where we're starting. I mentioned at the beginning, we're at the beginning of the beginning. And lastly, before I move to the next slide, we can and we will adapt our endpoints as the study is going on. And I'm going to show you a little bit of a schematic. I'm going to show you a schematic of what the study looks like, and I'll give you a little bit of an idea of where we'll be reevaluating the study endpoints as we proceed. All right, next, eligibility criteria. Who can participate in this study? And so I've outlined here a summary of some of the key eligibility criteria. I will mention that you can look at clinicaltrials.gov. You can find this study there. There's more specific data even on clinicaltrial.gov that's available for you to look at. So, and then of course, if you have any questions, I think eligibility criteria, a big topic for questions. You can speak to myself or Melissa Vigderson, who's pediatrician who's responsible for running this study at the back of the room. So feel free to speak to us afterwards. We've just tried to summarize the eligibility criteria here. To start off with eligibility criteria, I think it's really important for you to understand the why. So why do we select the eligibility criteria that we do? It's not because we're trying to determine who we want to have, like long term. It's not that we're deciding who we want to have access to the treatment. That's not it at all. So the objective here is to maximize the probability of detecting a treatment effect. So when you run a clinical study, there are two things that you want to make sure you do. One, if there's a treatment effect, you absolutely don't want to miss it, right? And that kind of goes to that benefit side of that balance beam I showed you. Two, you want to make sure that anytime you're exposing a patient to risk, you have a very strong rationale for why the patient could experience a greater benefit. So those are the two things that we keep coming back to when we design clinical studies. And when we look at eligibility criteria, the number one thing we're looking at is you want to start your clinical study in the population where if there's a treatment effect, you're absolutely going to see it and be able to characterize it. Another thing that's really important to understand is that eligibility criteria in clinical studies and during drug development always start very narrow, and over the course of the development program, they get broader. That is a trend that is pretty consistent in drug development. Especially in rare disease, I think I mentioned if you go to Bloomberg and you look at drug development programs, huge phase 3 studies that are being run for heart failure or psoriasis or diseases that are really, really common, you'll find that eligibility criteria of the phase 3 study often determine who can use the drug when it's marketed. That is not the case in rare disease. So I want to emphasize that in rare disease, and we have experience in rare disease, Melissa and I have worked on other rare disease programs. At Novartis, we've worked on many rare disease products. Eligibility criteria in studies does not equal the eventual labeling of the product for use. And so while we want to make sure that by the time a product comes to market, we've studied it in a broad population, there are limitations to how you can do that in rare disease with relatively few patients available to study. And so I think that's an important thing to point out also when we talk about eligibility criteria. Lastly, and this is something to be really clear about, the investigator, so the physician who's enrolling the patient in the clinical study is ultimately responsible for determining whether or not a patient meets eligibility criteria to participate in the study. So Novartis, as the sponsor of the study, we don't have any interaction with the patients. We don't decide who is eligible, who is not. That really is up to the physician who's participating in the clinical study as an investigator. So just kind of running through, that's a lot of background on why and how we determine eligibility criteria. Now I'll talk kind of about them specifically. So the first thing you see there is that we'll be studying patients that are between 2 and 5 years of age. And that's because we think this is the part of the population where we have the greatest opportunity to see an impact of the treatment on kidney function. You'll see that the patients, of course, have to have nephropathic cystinosis. They have to have been stabilized on cysteamine before entering the study. They have to have adequate kidney function, again, since one of our primary objectives is to assess effect of the treatment on kidney function. We want to make sure that their kidney function entering the study is good enough for there to be sensitivity to a treatment effect. And then so to kind of jump across to, they of course must have renal Fanconi syndrome, which is what we're attempting to treat. And then as you jump across to the key exclusion criteria, most of the exclusionary criteria have to do with the patient's baseline health status. So we need to make sure that these patients are healthy enough to be able to participate in a study like this, which involves really a very stressful process of having a hematopoietic stem cell transplant. Of the slide, of the study, of the system study. And before I talk through what the study is going to look like, I want to emphasize 3 things. So if you take away nothing else from this slide, take away these 3 things. So our study design is set up to, first, we want to be very cautious. So I talked about the fact that we want to make sure that every time we expose a patient, especially a patient between 2 and 5 years of age, to a risk, that we have very clear expectations that there's what we call prospect of benefit for that patient. Secondly, you've got to remember that this study is set up to be adaptable. So when we come back and we look at what's happened with this study 2 or 3 years from now, expect that you're going to see a very different study by that time. The study is going to change. And 3, we want to make sure that all of the data generated in this study is meaningful, meaning that we can interpret it and use it to support registration if the results of the study are overwhelmingly positive. Right? We know that there's a tremendous unmet need in cystinosis as a disease overall. We know that cystinosis is a very rare disease. We're not going to run a 500 patient outcome study to develop a cystinosis drug. There simply aren't enough patients. So we want to make sure that every patient that's treated, that the data that's generated from that treatment is usable. And I'm going to talk a little bit about how we've tried to do that. All right, so you can see that the study starts off with, there are actually 4 cohorts in this study. Cohort 1A, 1B, 1C, and then cohort 0. So I know some people, we've gotten some questions, people have looked on clinicaltrials.gov and said, there's 30 patients in the study. Well, 15 patients will be enrolled in, we're targeting enrolling 15 patients in cohort 0 and 15 patients overall in cohort 1. Now I mentioned that it's important for us that the data is meaningful, and so I'll actually start off talking about cohort 0, because cohort 0 is really, really important for us to ensure that the data generated by this study is meaningful. This is a standard of care reference arm. Patients who enroll in cohort 0, we will collect data from, but they will not undergo they will maintain their standard of care treatment on cysteamine, will not undergo the HSC therapy. So patients who are wanting to participate, wanting to be part of the system study, but not interested in participating in the active arm of the study, cohort 0 would be a great opportunity for them to be part of this. This data is important because comparison between patients that have been treated and this reference arm will give the data that we're generating in the treatment arm meaningfulness. And so it's a very important reference and was a very big part of our discussions with FDA on the meaningfulness of the data that we're generating. All right, now if we move up to the top of the slide and we talk about the first cohorts, you can see cohort A, we use the term sentinel dosing. So what that means is that we will be treating patients in cohort 1A, or the first patients in the study, sequentially, one at a time. And when we treat these patients, we will be assessing these patients for safety over a period of about 42 days, at which point they'll be, we have you can see it says there DMC, what that stands for is Data Monitoring Committee. So we'll have a committee of physicians who look at the data generated from those patients and make the decision, going back to the balance beam that we talked about, did we see any adverse events in this patient, in patient number 1, that would lead us to believe that the balance between benefit and risk is out of whack? If it is, we stop. If it's not, we move forward and we start patient number 2. Then after patient number 2, once again, they'll meet, they'll have that discussion and move forward with patient number 3. So this is an approach that's used in drug development really across the world. This is a very standard approach, and it ensures that we do the right thing for patients with respect to safety. Once the first 3 patients have been treated in cohort 1A, the Data Monitoring Committee will look at the data from those 3 patients overall, and if we still believe that the risks are as we expected, we'll then move forward into cohort 1B, where 3 patients can be treated simultaneously. And we'll be treating those patients, there won't have to be a one-at-a-time assessment. After those 3 patients are treated, we would then have another Data Monitoring Committee discussion, and here they would look at the data from the 6 patients all together and make the decision, now looking at 6 patients, when you look at 1 or 2, you're mostly looking at safety. When you look at 6, you're looking at safety, and then we've used a term there called futility. So we'll be looking at, is the product safe, and we should at that point have some early indication of efficacy, of benefit. And so it comes back to that balance beam again. It does benefit risk make sense to move forward with the study? And if it does, then we'll move forward into a much larger group, which we've called the expansion cohort. And we're hopeful that in the event that we make it to this point in the clinical study, the expansion cohort, we can design that in such a way that it would support registration of the product ultimately. Now, I mentioned several times during this talk that this study is designed to be adaptable. And I think it's important for you to understand that every time that we are looking at data from these patients, we're going to be reassessing both the design of the study, we'll be reassessing the study endpoints, we'll be reassessing the inclusion-exclusion criteria, right? So by breaking the study into 3 distinct cohorts, we put ourselves in a really good position to change any one of those cohorts depending on where we see the data and the science leading us. We also have the option to add cohorts, particularly when we look at cohort 1C, right? And so these are discussions that we're constantly having within the team. I'll also mention that because we will be starting 1 patient at a time, we'll start with a relatively small number of sites. And those sites will start off here in the US. By the time we get to cohort C, we haven't even determined what our sites would look like in cohort C. We'll be making decisions on whether or not we would go more broadly with those sites. All right, so last thing I'll mention here, and then I'm gonna just explain in the last 5 minutes what this is going to look like as we move forward. I want you to understand that cohort 1A is going to take a significant amount of time, right? So each patient is going to take several months to treat, and I think that's really important for the community to understand. So we're going as fast as we can, and the program will gain speed as it progresses because we'll be able to treat patients simultaneously and then more patients simultaneously. But initially, it's a relatively slow start. And the reason for that is that the HSCT process is not a fast one. Every patient that's treated will take quite a bit of time. And so I realize that we're fairly low on time, so I'm not gonna take too long walking through this, but I just want you to understand the process that every patient would have to go through for HSC treatment. We start off with stem cell mobilization. So once a patient's been enrolled in the study and they reach the point of stem cell mobilization, they're given some medicines that increase the fraction of their HSCs that are in their bloodstream. These are a specific type of HSCs called CD34 positive HSCs. We then collect those HSCs. We send part of those HSCs for manufacturing, and rescue aliquot is frozen. Those HSCs have to move in a relatively short period of time to a manufacturing site, and there they're treated with the lentiviral vector, and Stephanie talked through this step. Now I think it's important to note this is a really, really important step because not only do we have to manufacture the cells, so you take the patient's stem cells and you add the therapeutic transgene, but we also have to perform a lot of quality control testing because we want to make sure that before we reintroduce those cells to the patient, that they meet the right level of quality to ensure that they're likely to have an effect in that patient, right? And so we complete that manufacturing process and all of our quality control testing before the patient can move into the next step, which is myeloablative conditioning. And Natalie talked a little bit about her experience with myeloablative conditioning, where the patient's given a chemotherapeutic agent that effectively kills the hematopoietic stem cells in their bone marrow and creates space for engraftment of the genetically modified stem cells on transplant. And so this is a part of the process that involves a lot of risk for the patients. Natalie talked about her mouth sores. She talked about the impact of that chemotherapy on fertility. And so we want to make sure that we're 100% ready to go with the drug product before patients start that process. Lastly, after conditioning, the patient can be treated, and then it takes several weeks for hematopoietic reconstitution and then proliferation of those stem cells in the patient. Last slide before we go to conclusions here, I want to give you an idea of what this looks like for an individual patient. This is about a 3 to 4 month process from start to finish. So this is a really busy slide, but I think it's really important for you to understand that this is a big process with a lot of commitment for patients that want to participate in this study or have this therapy. I think before I walk through it, I want to point out these stars that we put on the slide and mention that there are multiple points during this process where we're going to confirm the patient's eligibility to ensure the patient's safety. So again, caution is one of the themes in our approach to this study. All right, so I'll just walk through. Screening of a patient will take, we think, roughly 2 weeks. Following screening, patients will go through a cysteamine washout of an additional 2 weeks where they'll stop taking their cysteamine. And following that, we'll have 1 week of baseline assessments. And this is really important because it allows us to capture data on the patient off of cysteamine and understand what their untreated baseline looks like. That's important because after the patients have been treated with the HSCT and are off cysteamine, we need a similar state to compare their data to pre-treatment. Following the cysteamine washout, patients will go back on cysteamine and they'll be evaluated again for eligibility before we go into this next step, which is the cell mobilization and apheresis step. And this is where patients go through the mobilization of CD34 cells. Apheresis is basically patients are go onto a machine kind of similar to a dialysis machine that effectively allows us to extract the CD34 stem cells from their bloodstream, concentrate those, and send them off to manufacturing. At the beginning, we expect manufacturing to take several months. The actual manufacturing of or not several months, we expect manufacturing to take about 2 months. The actual manufacturing process itself takes less than a week, but we spend several weeks working on the quality control of those cells to make sure that the cells contain the right amount of therapeutic transgene, and that when the cells begin to divide, that they're carrying that transgene. And that requires us to take those cells and actually expand small amounts of those cells. So put them into a dish, let them grow over weeks to make sure that we've got the right quality of product before reintroducing it to the patient. Once we're finished with all of that QC testing, we freeze down the product, and then the patient is cleared to go into conditioning with busulfan. Again, before they go into the conditioning step, there is a check for eligibility, and you can determine, we could actually decide to wait before conditioning a patient if their health status is not up at the level we need it to be to start that process. After conditioning, which takes several days, patients can then be treated with the HSC therapy. And at that point also the patients also will stop taking cysteamine, and that would then be followed by treatment